Good afternoon, everyone. We have seen yesterday the DNA and RNA, what are their structural differences and what are the features we are having for them, the different molecules and bonds and everything. And yesterday we discussed some biological functions of DNA and RNA. And now the application of DNA, we are going to have the important one is DNA fingerprinting. DNA fingerprinting. So what do you mean by fingerprint? I think everyone of you are having aware of that fingerprinting. So whenever we want to make any thumb impression, the recently what the NEET exam we are having, they are to take your thumb impression for making your identity. That is, whenever you are keeping your fingerprint, then the same fingerprint they are going to check it when you are going to write the exam. The person who applied and who is writing the exam are same or different. They can able to compare by looking at your fingerprint. That is what the fingerprint. How we are going to get the fingerprint? The fingerprint we are going to get it because of the protein structure what we are having on your palm. The palm what proteins are going to have, they are going to make an impression. That impression we are going to call as a fingerprint. That is the structure of protein what you are going to have on your hand. That is going to be taken as fingerprint. But these fingerprints are going to be altered whenever we are having some surgery or whenever you are undergoing some accidents. So during this time, the structure of your proteins on your palm may change. So before and after, see sometimes our phone lock only, we can't able to get open, even any cut is there on your finger. Isn't it? First when you are buying a phone, we are going to give the finger, so that is going to be registering. But whenever some cut is there, the same finger if you are keeping it, it can't be open because the fingerprint was altered when we are going to have a cut on your finger. So that's why the surgery can able to change the fingerprint. That's why the fingerprints are not unique. We can able to say they are going to be identified, but they are not unique. They can be altered for a surgery or by a particular any cut or damage is going to happen to your hand. That is the reason we are not able to believe the fingerprints during certain, say for example, criminal investigation done by a police people. They can't able to believe. Why? Because when he is doing that crime, that person is going to use his fingerprint, and after that he might have got a surgery on your hand and they can able to alter that. That's why we can't believe the fingerprints for the identification of a person. So we are going to see the DNA fingerprinting, which is going to be a new technique which can be very much useful for the criminals identification in a forensic studies and even paternity cases we are going to have. These are all the things can be solved by using DNA fingerprinting. What do you mean by DNA fingerprinting? How we are going to take the fingerprint of a victim and our suspective, and we are going to compare with what fingerprints we are going to get during a particular area of incident. That is what we are going to do in normal fingerprinting. In DNA fingerprinting, what we are going to do? So we will be collecting the samples from a particular person who is a suspectee, and we are going to compare whatever the DNA samples available where the incident was happened. Understand? So what is going to be taken here? Say for example some blood sample was they got in an incident place. So that blood sample will be analyzed, they will be collecting the DNA from that one, they will be making the DNA of that particular blood sample, and whoever suspect is there, from them they will be collecting the blood sample and they will be analyze the DNA, and they are going to check whether this DNA and DNA which is collected over there is same or not. Then there is no chance of having a different DNAs. We can't alter them. Always we can able to get exactly whether it is going to be same person or different person by looking at their DNA structure. So what we are going to identify DNA sequence? The sequence of nitrogen bases, what we are going to have, that is going to be compared with what the nitrogen base sequence we are having in a suspected person. So that if they are comparing, we can say 100% that person will be a criminal. Understand? We can able to make the DNA fingerprinting is a technique where we are going to compare the DNA of a suspectee with the DNA samples what we collected from the incident place. That DNA can be hair or nails or even blood sample or anything we can able to collect from them. Then we can able to take the particular DNA. That DNA is going to be compared with the DNA of the suspect. Understand? So these DNA fingerprinting exactly like the basis sequence is going to be same or not, we are going to identify by using this, and how we are going to perform this one. Say for example, you got some protein. That protein is nothing but sequence of amino acids. I collected a protein from there. The protein is nothing but amino acids. Each amino acid is coded by a triplet code. So this is going to be given mRNA. From this mRNA, we can able to develop the DNA. How we are going to do this one? DNA fingerprinting. We are going to take a protein sample. What we are going to get from the incident place. That protein is nothing but amino acids. We are having onboard amino acid analyzer. That analyzer is able to make the sequence of amino acids which are present in this protein. The sequence of amino acids will give us the idea about what RNA is synthesizing those amino acids based on genetic code. AUG is coding for methane. If first methane is there, we can able to construct here AUG. If we are having some other valine, glutamic acid, they are having certain codes. From there, we can able to synthesize the RNA, what is going to be synthesized in this particular amino acids. Then we can able to make the mRNA is nothing but sense strand of DNA we are going to get, and we can able to construct the complementary strand. We can able to get the DNA from this one. The same way, we are going to collect the suspected people and we will compare the DNA of the incident place and the DNA of suspectees. When they are going to be matching, we can say the person is going to be the particular criminal. And this is how we can able to use DNA fingerprinting in a discovery of criminals, and even we can able to use it for making the forensic lab just criminals only, where we are going to have paternity of individuals. What do you mean by paternity of individuals? So recently it is going to be a very passion, like I am the son of that particular celebrity, I am the daughter of that particular celebrity, like that so many are coming and they are reporting. So when we are going to have such cases, how can we be able to prove that person who is claiming is going to be really their son or daughter? That is going to be tested by using DNA fingerprinting. We are going to take the sample from the mother or father, and we are taking the sample of daughter or son. Then we are going to match their DNA samples. If they are having the matching, the DNA fingerprinting was going to be able to make either paternity or maternity cases what you are having, who you are claiming I am so and so, son of or daughter of some celebrity. That is going to be correct or not, we can able to say it by using DNA fingerprinting. And also we can able to make the things like what you can say dead bodies. Say for example, huge accidents were happening, bus was undergone accident, then the entire bodies are going to be get burned. We can't able to identify their faces, or we can't able to completely we can't able to say it. It will be like some flesh will be there. So then those flesh they can able to collect, and then they are going to make the DNA test sample for them, and the particular family member who is asking, yeah, my son is missing, my daughter is missing, like that, then they can able to make the samples of the dead bodies what they are having and what the sample their parents are giving. They can able to match their samples, and they are going to give the dead bodies to that particular person during accidents when happen. So these are all the usefulness of your DNA fingerprinting. We are going to have a wide range of applications are going to be developing by recent technologies what we are having, that is DNA fingerprinting. Understand? This is about your nucleic acids. And the last topic what we are going to have in this one is hormones. We are having hormones. What are hormones? messengers. I will call hormones as chemical messengers. How these chemical messengers are going to be produced? Actually, we are going to have two types of glands. What they are? Endocrine glands, exocrine glands. In our body, we are having two types of glands. All glands are going to have some secretions, isn't it? We are going to have so many glands in our body. All the glands are going to secrete something. These are going to be divided into two types: exocrine glands and endocrine glands. Exocrine glands, they are going to give you the secretions when they are going to be secreted into ducts. They are called as ducted glands. When we are going to take endocrine, so they are going to have no ducts. They are going to be giving you the glands are not having any ducts. They are called as ductless glands. So these ductless glands are going to be producing their secretions directly into the bloodstream, so that we can able to get directly the production and site of synthesis are going to be different for the hormone. Where they are produced, from there only they are going to travel to bloodstream, and where they have to act, they will be traveling through bloodstream and they will be acting. Site of production and site of action will be different for the chemical messengers which are produced by endocrine glands. Understand? So these are called ductless glands, these are called ducted glands. Now these ductless glands are going to be producing these hormones, they are called as chemical messengers. They are going to give you the information from intracellular to extracellular. Inside the cell to outside the cell, they are going to give, and that information is passing by these ones, that's they are called as chemical messengers. Anyway, what glands we are having? Pituitary gland is there, pancreas is there, and adrenal cortex, adrenal gland, and we are going to have thyroid gland. Like that, we are having so many glands. All the secretions they are secreting. Obviously, you are learning in your biology. But here, what is important for me, their chemical nature. How the hormones can be classified, and these hormones are made up of what chemical compound. That is important for our chemistry concern. Just we will go through that particular thing only. How the hormones are classified based on chemical nature. The chemical nature of hormones is going to give you total two types. Broadly we can classify. That is going to be steroid hormones, non-steroidal hormones. We are going to have steroidal hormones and non-steroidal hormones. There is the two types of hormones you can say, which are going to be derived from a steroid. Is called a steroid hormone. Which is derived from other than steroid, they are all comes under non-steroid. So the steroidal hormones, they can be classified into steroidal hormones. They can be classified into again two types. There are two types, sex hormones, and we can call them as corticosteroids. Steroid hormones are divided into two types, sex hormones and corticosteroids, and non-steroidal hormones are going to be divided into two types. Peptide hormones, amino acid derivatives. We are going to have two types, that is steroid, non-steroid. Steroid is of two types, sex hormones and corticosteroids. And in this we are going to have non-steroid two types, peptidal and amino acid derivatives. So now, what sex hormones we are going to have? The sex hormones are going to be mainly three types we are able to call: male sex hormones, female sex hormones, and we are going to have progesterones. We are going to have progesterones. So total 3 types of sex hormones we are going to have. These are going to be male sex hormones, female sex hormones, progesterones, or we can say pregnancy hormones. So the male sex hormones are going to be called as androgens. These are called as estrogens. Or I said these are pregnancy hormones or progesterones. Now coming to the male sex hormones, what they are going to do? The male sex hormones are going to give the secondary sexual characters. They are going to give secondary sexual character for a male. That is going to be the responsibility of this one. They are steroidal origin. And female sex hormones are going to develop secondary sexual characters in females. And we are going to have mainly estradiol here. Estrogens we are going to be. And here it is testosterone. And when you are going to take progesterones, they are going to be responsible for the pregnancy. During pregnancy, the nourishment of fetus and everything is going to be given by this one. It is produced by your what I can say uterus. This is going to be produced by ovary. Okay. So these are all comes under sex hormones. They are going to be present in a body. And coming to the corticosteroids, these corticosteroids are going to be produced from adrenal. And these corticosteroids are going to be, we can say 2 types, glucocorticoids, mineralocorticoids. These are going to be 2 types, glucocorticoids and mineralocorticoids. The name itself is going to influence, say the information regarding this one. Glucocorticoid is useful for maintaining the metabolism of glucose. It is going to be produced both of them by adrenal cortex only. Different cells are going to be producing. Glucocorticoids are going to be responsible for the metabolism of your glucose. Whereas mineralocorticoids are maintaining the mineral metabolism. And even NaCl, we are having the best mineral that is going to be get balanced by your mineralocorticoids. This is the role of glucocorticoids maintaining the carbohydrate metabolism. Mineralocorticoid is going to be responsible for the mineral metabolism. That is what we are going to have. This all comes under steroidal hormones. Coming to the non-steroidal hormones. Non-steroidal hormones, we are thinking, we are having so many peptidal hormones are there. Out of these peptidal hormones, we can say the important ones are insulin. I think everyone knows about this insulin. What is insulin? It is going to be a hormone secreted by your pancreas. Here we are going to have alpha cells and beta cells of Langerhans. So they are going to be producing insulin and glucagon. Both of them are having same origin, that is they are of derivatives of peptides. They are all made up of proteins. The insulin hormone is going to have 2 chains, A and B. A chain is going to have 21 amino acid, B chain is going to have 30 amino acid. These 2 chains are going to be combined with a disulfide bridges. So that is what the structure peculiarities we are having for insulin hormone. What this insulin hormone is going to be called as hypoglycemic. Overall effect is hypoglycemic effect. Whereas glucagon is going to show hyperglycemic effect. Glycemia means which is going to be increasing the glucose level in blood. Increasing the glucose level in blood is nothing but glycemia. Whereas hyper means increasing, hypo means decreasing. The glycose or glucose in blood is called glycemia. If it is hypo, lesser. Hyper, it is higher. That is what you are going to have a hypoglycemic effect and hyperglycemic effect. These 2 hormones are going to be, both of them are secreting from pancreas only. Both of them are producing from pancreas only. Other than this one, we can able to say vasopressin. Why I am discussing vasopressin here? Insulin deficiency causes Diabetes mellitus. This deficiency causes diabetes mellitus. Whereas vasopressin is going to be cause diabetes insipidus. It is called diuria. That is more urine secretion. Vasopressin is anti-diuretic factor. It is going to be deficiency causes diuria. That is more urine secretion. Whereas diabetes mellitus is going to be sugar, a very popular disease we are going to have nowadays. After 40, everyone is going to get insulin because of lack of insulin. So this insulin deficiency is going to cause diabetes mellitus. Even hereditary also we are going to have diabetes mellitus. That's right. We are having anyway the chance of getting sugar is common nowadays. We are supposed to be careful about it. Other than these hormones, we are having so many. Like all your peptide, pituitary hormones are comes under this one only. We are having all pituitary hormones comes under peptidal hormones only. This is about the peptidal hormones. And coming to amino acid derivatives, a very important thyroxine. T3 and T4. T3 and T4, which is going to be a hormone which is secreted from thyroid gland. Both T3 and T4. T3 is active, T4 is less active. T3 hormone is going to be derivative of your aromatic amino acid tyrosine. This is going to be a tyrosine derivative. This tyrosine derivative, thyroxine, is going to be very important hormone secreted from thyroid gland. And this hormone is responsible for your lipid metabolism mainly, or activity of body, BMR of the body is maintained by your T3 hormone. Deficiency of this one is going to cause hypothyroidism, and if you are having secretion of more, it's called hyperthyroidism. Hypothyroidism leads to obesity, hyperthyroidism leads to lean. So both are going to be dangerous. But so many people, mainly female, are going to be affected by hypothyroidism, which is leading to the disturbances for the menstrual cycle, so that there is a problem in getting a pregnancy. That is what we are having. Thyroid care centers are so many nowadays. That is because of this thyroxine hormone. That level of hormone should be maintained properly. Otherwise, any imbalance is going to cause either hypothyroidism or hyperthyroidism. Okay, so this is overall we are supposed to remember this table where we are going to have the information regarding what hormone is going to have what nature. The chemical nature is going to be steroid, non-steroid. Steroidal hormones are two types, sex hormones and corticosteroids. Androgens are male. It is going to be testosterone, which is developing secondary sexual characters in case of male. Female are estrogens. This is going to be developing the secondary sexual characters in female. Progesterones are going to be produced by uterus. They are going to be nourishing the fetus during the pregnancy time. Corticosteroids are two types, glucocorticosteroid and mineralocorticosteroid. Glucocorticoids are maintaining the metabolism of glucose, whereas mineralocorticoids are maintaining the metabolism of minerals, mainly the sodium and chloride we are going to have. Non-steroidal hormones are two types, peptidal and amino acid derivatives. Peptidal hormones are going to be main important ones are insulin, glucagon. Insulin is going to be showing hypoglycemic effect. It decreases the glucose level in your blood so that we can able to maintain the glucose properly in the blood. Whereas glucagon, what insulin will do when insulin hormone is secreted, the excess of glucose, anything is there, it is getting converted to glycogen. If we are having low glucose levels, then glucagon will be secreted. It is going to make the glycogen converting into glucose. That is what the opposite action we are going to have between these two. The insulin deficiency causes diabetes mellitus, and vasopressin deficiency causes diabetes insipidus, which is going to be called as anti-diuretic hormone. Whereas amino acid derivatives are having thyroxine, T3, T4, which are going to be causing hypothyroidism or hyperthyroidism, which is going to lead more disturbances for the total body metabolic rate. So that is not supposed to be happen. We are supposed to take care of this T3, T4. Even iodine deficiency is going to cause hypothyroidism. we are not taking the iodine properly, that is obviously going to cause hypothyroidism. That is what you are going to have from this one. And this is what the biomolecules chapter. I hope everyone can able to understand whatever we are discussing here. If any doubts are there, you can able to ask me during the interaction time. Anyway, when you are coming here, we are going to explain you when we are having the doubts. Okay, so this is the end of your biomolecule chapter. The next chapter we are going to discuss is chemistry in everyday life.